Hello, everybody. We'll just wait for 2-3 minutes and then we will start the session, okay? Okay, so we will start the class now. So, good evening, everybody. Today we'll discuss the lectures and the questions related to week 9 of our lecture. So, week 9 was about dosage compensation and X inactivation. Okay, in this week there were 4 or 5 lectures, and this is a very important topic. This is an important topic not just in relation with RNA biology course, but in general if you're going to give any competitive examinations or you are appearing for any other kind of examinations, this topic aligns with not just RNA biology, this topics aligns with many other subjects as well. So this is an important topic. Okay, so I hope that you have studied a little bit about this topic, and I think you must have studied about this in your like some other subjects or previously as well, because it's a well known and a well established topic. Okay, so let us start. So just a minute. Yeah. Our first lecture was different modes of SRP and different modes of compensation. Okay, so what is the meaning of SRP? SRP means signal recognition particle. Okay, SRP means signal recognition particle. So this SRP is composed of both proteins and it is composed of RNA also. And you can see the structure of SRP. You can see the structure of this signal recognition particle. Um, just a minute. Yeah, this is your RNA. Okay, this whole area that you can see, this all is your RNA. And RNA holds together the protein subunits of SRP as well as it helps them to bind to the ribosome. Okay, you can see RNA, what they are saying, that it holds the protein subunits of SRP. See, this is your SRP. So basically RNA is holding the protein subunits of SRP also. Also it helps and it interacts with the ribosome. So both the functions are performed by SRP. Now then we come to tmRNA and translation. What is tmRNA? tmRNA is basically a hybrid molecule which is half tRNA and half mRNA. Okay, so this is a hybrid molecule, tmRNA. It's a hybrid molecule which is half tRNA and half mRNA. And tmRNA helps to rescue ribosomes bound to mRNA which lacks the termination codon. So suppose if this is an mRNA and this is the ribosome which is attached to the mRNA. Okay. Now this ribosome, as soon as the termination codon comes, what happens? As soon as the termination codon comes, the ribosome is detached from the mRNA and your translation stops. But sometimes, if RNA does not have any termination codon, because there is no termination codon, then ribosome will not get any signal to get detached from it. So then in that situation, in that case, what is the way by which this ribosome can be helped? So basically, tmRNA comes into rescue in that picture. Okay. So what it does is that because it's a hybrid molecule, it is half tRNA and half mRNA. Basically, it can help to detach this ribosome from the mRNA. Okay. So this tmRNA will help to detach the ribosome from the mRNA. So this is why tmRNA, T for tRNA, M for mRNA. Okay. So this is what tmRNA stands for. So here is the mechanism of how the tmRNA helps in this process. So tmRNA has tRNA portion also and mRNA portion also, and that is the reason why it is known as tmRNA. So what is written over here? The tRNA portion of the tmRNA carries an amino acid alanine, whereas mRNA has a bunch of codon and also has stop codons. Okay, this is fine. So what happens if the termination codon by some mistake is not reached? The ribosome gets stuck upon reaching the 3' end and has to be rescued. Since the stop codon is not reached, the newly made protein is probably wrong and it needs to be degraded. Okay. The tRNA part adds an alanine to the growing polypeptide chain, and the mRNA part enters the ribosome, and the synthesis of polypeptide is continued with the aid of normal tRNAs until the termination codon is reached. And in the end, ribosome is released and the newly made fusion protein is degraded due to signal sequence. So basically what happens that suppose if this is your mRNA and your ribosome could, there can be two reasons. First, there is no stop codon, and if there is no stop codon, that means there has been some fault or there has been some problem in the mRNA. And since there has been some fault and problem in the mRNA, this particular mRNA, first the ribosome has to be removed, and secondly this mRNA needs to be degraded. So in that case, your tmRNA comes into rescue. Not only it removes or detaches the ribosome from the mRNA, but it also degrades that mRNA. Second situation can be that suppose this is the ribosome and the termination codon is somewhere here, but the ribosome couldn't reach up till the termination codon, and this is the 5' end and this is the 3' end. Towards the 3' end, the ribosome has got stuck. Now it cannot go further and it cannot reach the termination codon. So in this case, this ribosome has to be rescued. So what happens? And also because the stop codon is not reached up till only, suppose this is the stop codon. Okay. Here your stop codon is there. Your ribosome has now attached at this place and now it is stalled here. Stalled here means now it cannot move away from here. So now what happens? This ribosome is good only, but there is some problem in the mRNA coding sequence. So this ribosome has to be detached from it. So in that case, what happens? That tmRNA comes to the rescue. Not only this ribosome has to be detached, but also this mRNA should be degraded. Why? Because since it could only synthesize up till here, it hasn't it hasn't translated the whole sequence, so it is not a complete protein that is formed. Okay, so which means that this protein is not a complete protein, and since this protein is not a complete protein, it should be degraded because it is not fully functional. This protein, whatever protein will form, will not be a fully functional protein. Okay, so what happens? This tmRNA, it has a tRNA part and it has mRNA part. tRNA part, what it does is it adds alanine to the growing peptide chain. And mRNA part enters the ribosome and the synthesis of polypeptide is continued up till the termination codon is reached. So in the end, what happens? Ribosome is released and the protein is degraded. So this is just a little bit about the tmRNA portion before we go to the main portion. Now, since we are studying about this dosage compensation, how it comes here, we need to see that. So what happens that we were studying about the non-coding RNAs. Okay, we were studying about the non-coding RNAs. So non-coding RNAs are the RNA which does not code for the protein, but they do have some functions. Now, what function these non-coding RNAs can have? Just a minute, there is some problem it seems. बेबी एक बहुत बड़ा सा मच्छर था, वो मेरा खून चूस रहा था, हमने उसको मारा तो मेरा खून मेरे मुंह पे लग दिखा फोड़ दो इसमें फोड़ दो रगड़ दो अच्छे से कहाँ लगा मुंह पे हाँ ठीक है मैं कौन कर लेता है yeah. So, non-coding RNAs have many functions and they have many regulatory roles. So, what are the regulatory roles of non-coding RNAs? They can be transcriptional regulators, they can be translational regulators, they can be modulators of protein function, and they can be regulators of RNA and protein distribution. So, there can be many roles, regulatory roles of non-coding RNA. Okay, they help in regulating the process of transcription, translation, the protein function, the distribution of proteins, and many more. But one of the most important function of these regulatory RNAs is dosage compensation. Okay, so what is the meaning of this dosage compensation? Let us try to understand this. So, talking about humans, we have females and we have males. So here we have XX. Two X chromosomes are there, okay? And in male, we have X and Y chromosome. That means here the number of X is more, here the number of X is less. So now to make sure or to ensure that X-linked traits are equalized between females and males, one of the X chromosome is inactivated in the case of females. And this inactivated X chromosome is known as bar body. Okay, this inactivated X chromosome is known as bar body. So that means the dose of X should be equal in males and females both. So that means there has to be some mechanism which ensures that X linked, the amount of X remains the same. The traits which are carried through X remains the same. The dosage compensation should be there. So there are certain mechanisms by which it happens. Okay, there are various mechanisms by which dosage compensation can happen in various across various species. Fine. So let us try to understand what are those mechanisms. So you see, we have mammals, we have C. elegans, and we have Drosophila. Okay. So let us try to see what are the different mechanisms that happens in mammals, C. elegans, and Drosophila. So these are males. Males have XY and female have XX. So now males have only one X and female have two X. So that this means whatever the traits are coming, that can come from two of the X's, but here only one X is there. So only one X should come ultimately. So one of the X gets inactivated. So this X is inactivated now. This X is inactivated. So since this X is inactivated now, it is equalized. One X each from male, one X from female. In the case of C. elegans, what happens is that there is repression. So what is the meaning of repression? This is X. Suppose this is X1. This suppose let us try to understand in this way. Just a minute. Hmm. Let us assume this X as 1 for males. In females, you have 1 plus 1. So it would be 2. So will there be dosage compensation? No. What if I repress it and make it just the half of itself? Uh, just a minute. Hmm. So just to quickly recap that we are studying about the dosage compensation mechanisms across mammals, C. elegans and Drosophila. So we have discussed that in mammals you can see that one of the X chromosome gets inactivated. Okay, so what I'll do is that I'll try to make you understand with any arbitrary unit which has no numbers. So when I talk about mammals, okay, so females have XX and males have XY. So what happens? Suppose if this is 1 and this is also 1 and this is also 1. Okay, I'm just taking this arbitrary unit to make you understand how it happens. Okay, so what happens? It is 2 here, it is 1 here. So what happens? One of the X chromosome is inactivated. Since one of the X chromosome is inactivated, now it is only 1 and here it is 1. So compensation is done. Then we come to next, which is C. elegans. C. elegans, females XX, males XO. Am I writing it correctly? Just a minute. Yes. Here what is happening? Here repression happens. Here repression happens. So what happens here? If this is 1 and this is also 1, suppose, and this is also 1, so this would be 1 plus 1 would be 2 and it is here 1, so it would be not equal. What if this is repressed to half? So it will be both the x's are there. Here one of the x was inactivated. Here none of the x's are inactivated, but they are repressed to express by their half. So what happens? It is half plus half and 1. So it is 1 is equals to 1. So here also the compensation has happened. So here one of the x is inactivated. Here both of the x's are repressed. So that again it becomes equal. And then we come to Drosophila. Okay. Drosophila. So in Drosophila what happens? Males are XY and females are XX. Okay. Females are XX and males are XY. And what happens here is hyperactivation. Hyperactivation. Okay. So what is the meaning of hyperactivation? This is X1, 1. This will make 2. Okay. This 1 plus 1 will make 2. And this is 1 only. So what happens? Nothing happens to here. This X becomes hyperactivated. So if this is 1 plus 1, it becomes hyperactivated and it becomes 2. So again, so here X chromosome inactivation, repression of both the X chromosome, hyperactivation of the male's X chromosome. So could you understand? These are the different mechanisms of dosage compensation. The mechanisms may be different, but the ultimate goal is the same. It should be equal. The amount of X should be equal in your males and in females. That is the main objective here. So I hope this is clear to you. This is very important. That's why I gave so much of time in explaining the various compensation mechanisms across mammalians, C. elegans, and Drosophila. Okay. So now let us see dosage compensation of sex chromosomes. To equalize the dosage of X chromosome gene products by means of inactivating one of female X chromosome in mammals. Yes, in human females, XX1 chromosome is inactivated, the bar body. In Drosophila males, double the expression of genes along X, yes, hyperactivation. In C. elegans, both X chromosomes are partially repressed, repression. Okay, so now this the question should come in your mind that in the case of humans or mammalians, one of the X chromosome is inactivated. So what happens to that inactivated X chromosome and how it is found in the body, how it like looks like, as in molecularly, what are the characteristics of this X chromosome, that you should know. So this X chromosome inactivation is also known as lionization. Okay, this X chromosome inactivation is also known as lionization, and this X chromosome is maintained in the form of bar bodies. Okay, so this bar body is nothing but an inactivated X chromosome, condensed, darkly stained structure that is found in most cells of female placental mammals. So it is condensed and darkly stained structure. Why? Because it is inactivated. It is transcriptionally inactivated. So since it is transcriptionally inactivated, it will mostly have what? Heterochromatin. Okay. So since it will have mostly heterochromatin, it will be darkly stained and it is condensed. So any inactivated region of your chromosome is always darkly stained and condensed. Why? Because darkly stained because it is mostly heterochromatin, and it is condensed because, see, if your chromosome is just condensed like this, there is no place for the transcription factors to come, sit, and start the transcription process or start the further expression process. So, for in order for a chromosome to be transcriptionally activated, it should not be in a very condensed form. It should be loosened up so that there is a place for the transcription factors to come and bind and start the transcription process. So whichever area is inactive, it is condensed and it is darkly stained. Okay, so this is your bar body. So this is your condensed. Bar body is condensed and it is a darkly stained structure. Fine. So now we comes the role of RNA. What is the role of RNA here? Since we are studying about RNA biology, there should be some relation with RNA because we are studying about RNA biology course. So, this X chromosome inactivation that we are talking about, this happens with the help of a non-coding RNA. We just have studied that one of the regulatory roles of non-coding RNA is in dosage compensation. We have studied about the different mechanisms of dosage compensation across humans, mammalians, C. elegans, and Drosophila. Okay, so now what happens is that this X chromosome inactivation that happens, it is mediated by a long non-coding RNA which is known as XIST, XIST. Okay, so this X chromosome inactivation is because of a long non-coding RNA, and this LNC RNA is cyst. Okay, this is, which is a 16,000 nucleotide long non-coding RNA. Okay, now this cyst non-coding RNA recruited complex has one entry site in the X chromosome corresponding to cyst gene itself. So basically what happens that it binds to the X chromosome and cyst appears to recruit a specific histone isoform which is H2A1.2, which maintains the chromatin in the chromosome in the inactive state. So whatever I am marking are the important points. This is also important. This is also important. And this is also important. Okay, so I have marked all the important points over here. So what is there? Cyst appears to recruit a specific histone isoform which is H2A1.2. So what happens that this cyst, it binds to the X chromosome and then it recruits a histone isoform which is H2A1.2. Now this H2A1.2 maintains the chromosome in the inactive state. Okay. So since this H2A1.2 is maintaining the chromosome in the inactive state, your X chromosome gets inactivated. Also, this cyst complexes, they also recruit histone deacetylases and methylases. What is the meaning of histone deacetylases and methylases? These are the enzymes which will make the chromosome transcriptionally inactive. Why? Because two things are there in general. You should understand this, that acetylation and methylation are the two processes that happens. Okay. So, just a minute. Hmm. Acetylation and methylation are the two processes that happens. Acetylation m- makes your chromosome transcriptionally active. Transcriptionally active. It is because of acetylation. And methylation, it makes transcriptionally inactive. Okay. Methylation. So if it is, what is the opposite of acetylation? Deacetylation. What is the opposite of methylation? Demethylation. So if acetylation promotes the transcription, then deacetylation will repress transcription, and methylation is anyways repressing transcription. So which means that deacetylases will also means deacetylases will repress transcription, and methylases also represses transcription. So basically zist, it recruits these histone deacetylases and methylases so that it is completely made inactive. When I say that a chromosome is inactive, what does it mean? It means that it cannot undergo the transcription process. Okay, inactive or active in the case of chromosome means that it cannot undergo its expression. Activity and inactivity is in regards of its expression. Whether it is transcriptionally active or whether it is transcriptionally inactive, it means whether it can express itself or not. So X chromosome is inactive. When I say, it means that it cannot express itself. That is the meaning of its active or inactive state. Okay, transcriptionally active or transcriptionally inactive. So this is it. Then zist activity is regulated. Now this is what zist does. So zist, it binds to the X chromosome. Where it binds, there is a cyst gene. There is a cyst gene itself which is present on the X chromosome. It goes and it binds to there, and then it recruits a specific histone isoform which is H2A 1.2. Now this H2A 1.2 maintains the chromosome in the inactive stage. One thing. Second thing is that this also recruit histone deacetylases and methylases, which again maintains the chromosome in its inactivated stage. So in this way, this zist helps in silencing of one of the X chromosomes. Now the activity of zist, which is a long non-coding RNA, is also regulated, and the activity of zist is regulated by another long non-coding RNA, which is known as TXIS. T6. T6. This T6 has antisense sequence of zist, and therefore it is able to regulate zist activity by base pairing. So whenever this zist activity is needed, zist is there. Whenever zist activity is not needed, then T6 comes. Okay, so because not every time we want this activity to take place, only at specific times we want this activity to take place. So whenever we want the activity, it happens. When we don't want the activity, then T6 comes, and T6 again is a long non-coding RNA which is an antisense sequence to zist. So it comes, it binds to zist, and it regulates its activity. So I hope this much is clear to you. Okay, then this is what the mechanism that we have just now discussed. That zist expression is followed by irreversible layers of chromatin modifications that includes a loss of histone acetylation and this methylation that are associated with active chromatin state, and the induction of repressive chromatin modification includes H4 hypoacetylation, H3K27 trimethylation, H3K9 hypermethylation, H4K20 monomethylation, as well as H2A K119 monoubiquitination. Okay. Now we come to next thing is omniprinted versus random X inactivation. Okay. Now we have studied that in mammals, what happens? Humans, there is X chromosome inactivation that happens for to maintain the doses compensation. Okay, so this X chromosome inactivation, uh, what it does is that it inactivates one of the X chromosome. So the amount of X are equal in males and females. How it does it? It does it with the help of a long non-coding RNA, which is known as ZIST. So what this ZIST do is that it binds to the X chromosome within its ZIST gene, and then it recruits some histone, which are H2A 1.2, some histone isoforms it recruits. Also, it brings some complexes like histone deacetylases and methylases, and ultimately they all maintain your chromosome in the inactivated state. This is clear. Now, next question that should come in your mind is that this X chromosome inactivation that happens, whether it happens randomly or there is some printing, genomic printing that happens for this X chromosome inactivation, that it will happen in males or it will happen in females or it will happen in certain stage or whatever it is. So you should know that there X chromosome in X chromosome inactivation, it can be random also, it can be imprinted also. Okay, but imprinted X chromosome happens only at specific stages. It's not that always or at every stage imprinted X chromosome inactivation happens. No, it happens at certain stages. What are those stages? So basically, it happens in trophectoderm and primitive endoderm of the blastocyst. And here, what kind of imprinting is there? X inactivation is subjected to imprinting of paternal X chromosome. That means the paternal X chromosome inactivation happens. And epiblast stage X chromosome inactivation is random. Okay. So this is, you should understand that X chromosome ka inactivation, it can be, mostly it is random. It can also be imprinted, but it is imprinted only at specific stages, which is the trophectoderm and primitive endoderm of the blastocyst. So here we come to study about the random X chromosome inactivation. And to study the random X chromosome inactivation, we have a very good example of calico cats. Okay, calico cats. These calico cats are almost always females. So what happens over here is that coat color, the color of their coat, it has two alleles. Okay, it is XB and XB. This stands for orange coat. Okay, this one stands for orange and this stands for black. And this orange is dominant over black color. Okay, so when it is like this, XB, XB, it is obviously, what would be the coat color? Orange. If it is XB, XB, what should be the coat color? Black. And if it is heterozygous condition, XB, XB, then what should be the coat color? So logically, it should be orange. Why? Why it should be orange? Because orange is dominant over black. But this is not the case that happens in reality. What happens that the heterozygous cat with X capital B and X small b shows color patches due to random inactivation of X chromosome in different cells of blastula. So what happens is that the X chromosome inactivation is random. And since the X chromosome inactivation is random, we don't know, some places this particular black is inactivated, some places this is inactivated. So where this dominant one would be inactivated, the color would be black. Where this small one would be inactivated, the color would be uh, uh, orange. So we don't know, because we don't know ki whether the dominant allele is getting inactivated or the recessive allele is getting inactivated, because it is random. It is not uniform that in each and every cell only dominant will get inactivated or only recessive will get inactivated, because it is random. So there are patches. Okay, you can see here there are patches. There are patches because X chromosome ka inactivation is random. At some places, this particular X B is getting inactivated. At other places, this particular B is getting inactivated. We don't know. So this is an example of calico cat, which shows that the inactivation of X chromosome is random. Is it clear to everybody? Am I making sense? I hope this is you're able to understand this. Okay. Just go through this figure, 2 minutes. Just read it. Okay. So I hope this is clear. Now we come to the next thing, which is the molecular basis of X chromosome inactivation. Now that we have studied the mechanism of how X chromosome inactivation takes place, we have also studied that this X chromosome is mostly is random, and in certain stages of protectoderm and epiblast, it, it is non-random. This all we have studied. So now we come to the molecular basis. So in the molecular basis, this particular is clear that profectoderm and primitive endoderm, it is there is imprinting. Epiblast cells, there is X chromosome inactivation is random, and you can see that how this T6 is basically a regulator of the zist. So how this T6 regulate the zist, this is all is given over here. Okay, this is all the this all just describes that how inactivation takes place. Just go through it for 2 minutes. So what they are writing over here is the X chromosome inactivation center. So basically, now we will be studying about X chromosome inactivation center. So increase in stability of this transcript is brought by developmentally regulated switch in promoter use. So P1 and P2 are used for transcription of stable cyst, and P naught is used for unstable cyst transcription. Okay. And hyperacetylation disappears upon differentiation, and stabilization of cyst RNA might depends on the levels of cyst RNA present. So how your T6 and cyst are regulating each other, this is what is explained in this figure. Okay. Okay. So next is X chromosome inactivation center. So again, they are describing the same thing, that how it happens. This is something that we have just discussed. This is all describing that how this happens. Okay. So T6. T6. What is T6? T6 basically it regulates your cyst. Okay. T6, it regulates cyst. So this T6 is transcribed from antisense strand, which is related to cyst. In undifferentiated cells, T6 is expressed on both axes. In differentiated cells, it is only present in XA, and it prevents the upregulation of cyst on XAES. So it influences X chromosome choice and imprinting. So basically, it is regulating your cyst protein. Now, X inactivation, XIC, X chromosome inactivation center, locus on X chromosome. So basically, what happens that on X chromosome there is a locus which is known as XIC. This is a locus, means a particular area within the X chromosome. So this is a locus, and it contains non-coding RNA such as ZIST, T6, JPX, and FTX. So all the non-coding RNAs which are related to your dosage compensation, they are confined and present at one place, which is known as X chromosome inactivation center. What happens is that ZIST gene transcribes a long non-coding RNA ZIST, and ZIST RNA is spliced polyadenylated, but it is not translated. So basically, for the X inactivation, there is a certain defined, regulated, dedicated space that is present. Okay, which is your XIC, which is X chromosome inactivation center. Okay, I hope this is clear to everybody. Now, now we come to the X chromosome inactivation and RNA interference. So let us try to understand it from here. In mammals, females carry two X chromosome, whereas males carry only single X chromosome, giving a double dosage of X-linked genes in XX related to XY males. Yes, we have studied it. We are just revising it. So to balance X-linked expressions levels in males and females, female cells involve a mechanism which is known as X chromosome inactivation to randomly silence one of the maternal X chromosome during early embryogenesis. Okay, this we have studied. So what happens that prior to cell differentiation, both the X chromosomes are active. You should remember this. This is important. XA means activated X. Means so prior to cell differentiation, both the chromosomes are active. During cell differentiation, what happens? Each cell counts its number of X chromosome. Okay. Then one X chromosome is randomly selected for inactivation. The LNC RNA, which is ZIST, is upregulated on the future inactive X. Okay, so whichever X which is marked for inactivation, in that particular X, your ZIST is upregulated. Okay, suppose if there are two X now, and suppose randomly this X has been selected for inactivation. So in this particular X only, this ZIST would get more activated. I hope this is clear. Then a gradual chromosome wide silencing process is then inactivated on this particular X chromosome. Once established, this silent state is stably transmitted through each round of cell division in a heritable manner. Suppose if this particular X has been selected, then what happens? Then now it is this X chromosome has been selected, and now there is a in a heritable manner this X is used. I mean, this X will be silenced in further generations for further generations also. Could you understand this? Once established, this silent X state is stably transmitted through each round of cell division in a heritable manner. This is what is. Now, during development, females undergo two form of X chromosome inactivation. This is also we have talked about. So, X chromosome inactivation can be either random or it can be imprinted. So, imprinted X chromosome inactivation occurs during early embryogenesis. The paternal X chromosome is preferentially silenced. So, imprinted X chromosome only happens during early embryogenesis. Early. Just a minute. Okay. Yeah. So, basically, what happens that this imprinting of the X chromosome, imprinted X chromosome inactivation, it happens during the early embryogenesis. And random, all imprinted epigenetic marks on XP are erased in cells of the epiblast lineage. A second round of X chromosome inactivation is then initiated, whether either XP or paternal, paternal X chromosome or maternal X chromosome is randomly silenced. Okay, this is your random X chromosome inactivation. Now, X chromosome inactivation is initiated from the X chromosome inactivation center, which is known as XIC, which encodes 5 non-coding RNAs of known function, which is ZIST, T6, ZITE, REP-A, and JPX. ZIST is upregulated only after transient pairing of 2 X chromosomes, and it is the way, okay, uh, and then your depletion of any of the factors which are involved in pairing will lead to cells with aberrant number of active or inactive chromosomes. Okay, so this is the summary, and then I'll come to the questions. So in summary, what we can say, just a minute. Yeah, fine. X chromosome silencing via spatial and temporal compartmentalization. Okay. In perinuclear space, X inactivated X and other heterochromatin could replicate. Means if this is the nucleus, this is the nucleus, and this is the periphery. So your heterochromatin is at the periphery. Your transcriptionally active materials are present at the center. Transcriptionally inactive materials are present on the periphery. So in this perinuclear space, heterochromatin is present and your inactivated X chromosome is present. And whatever euchromatin or whatever the X which are activated are present in the center of the nucleus. So that is why it is saying that X chromosome silencing happens via spatial and temporal compartmentalization. So whenever we say compartmentalization, I don't know whether you know about this concept or not, but there is a compartmentalization of nucleus which takes place, and the two compartments are known as compartment A and compartment B. They are also known as permissive or repressive. Permissive compartment or repressive compartment. Okay, permissive compartment is the compartment which is present at the center, which contains all the activated genes, activated materials, what all which is transcriptionally active is present in the permissive compartment in the center. Okay, and repressive compartment is present on the periphery, which has all the heterochromatin things, which are repressive, which are not going to be transcriptionally active. So this is always the case. All the active materials are present in the center. All the inactive materials are present in the periphery. And this is known as compartmentalization of the nucleus. Spatial compartmentalization of the nucleus. Now, that is why it is written that in perinuclear space, this is the perinuclear space, the inactivated X chromosome and other heterochromatin is present. Now, cyst expression targets autosomes to the perinuclear spaces. Cyst deletion will lead to loss of heterochromatin, transcription upregulation. And this is all the questions for you to further understand. That is okay. So, with this, we have completed the theory. Now, we'll move to the questions which are related to this particular topic. So, I hope that up till now it was clear to you. I mean, the theory was clear to you. I have tried to explain the theory because mostly in this particular lecture, it is mostly about the X chromosome inactivation, how X chromosome inactivation take place. Since we are studying about RNA biology, what is the role of RNA in this X chromosome inactivation? So what we have studied, what is the role of RNA in this X chromosome inactivation? Basically, it is the long non-coding RNA which has a role in X chromosome inactivation. This long non-coding RNA, which is known as XIST, this ZIST basically goes and bind to that particular X which needs to be silenced, which need to be inactivated. So XIST will bind to that uh, going to be inactivated X chromosome only, and this ZIST will ultimately what will it do? This ZIST will then uh, increase the expression in that particular X chromosome, and then what it will do, then it will recruit some histone isoform like H2A1.2, which maintains or which causes the chromosome to be in the transcriptionally inactive state. And then it also recruits other some enzymes like deacetylases and methylases, which maintains the chromosome in the inactivated stage. So this is what we have studied, why there is a need of X chromosome inactivation, what is the difference between random and imprinted, at what stages does the random and imprinted X chromosome inactivation happens, and what are the different mechanisms of X chromosome inactivation that happens across various species such as Drosophila, C. elegans, and your mammalians. Now we'll come to the question. So how we do the questions? Always that I'll put the question on your screen. I'll give you 2 minutes to answer the question, and after that I will switch up the screen. So the first question is, histone unique to bar body is H2A 1.2, H3A, H1B, H4A. So which histone is unique to the bar body? Giving you 2 minutes, think, and then try to answer this question, okay? Think what can be the answer. We have already dealt it. Okay, now I am switching the screen. Let us see. Hmm. So the question is, histone unique to bar body is H2A 1.2, H3A, H1B, and H4A. So what is the correct answer? The correct answer is H2A 1.2. We have already studied about this. What we have studied? That ZIST is the long non-coding RNA. Long non-coding RNA. Non-coding habitat is LNC RNA. Long non-coding RNA, which goes and it binds to the X chromosome in the ZIST gene which is present. And then it recruits histone isoforms. And what is that histone isoform? This histone isoform is H2A 1.2. And when this histone isoform H2A 1.2 is recruited, it makes the chromosome, it helps in mediating the chromosome. It mediates the chromosome to be in the inactivated stage. So the correct answer is H2A 1.2. I hope this is clear. Okay. Next question is, ZIST influence the X chromosome A randomly, B non randomly, C irregularly, or D exclusively? What is the correct answer? How ZIST influences the X chromosome? Right now it is 7:54. I will switch the screen at 7:56. Okay. Think we have studied about it in a very detailed way. 1 more minute left. Okay. Now I am switching the screen. So, cyst influence the X chromosome randomly, non-randomly, irregularly, or exclusively. So, what is the correct answer? Cyst influences the X chromosome exclusively. How it influences the X chromosome exclusively? Because there are X chromosome is there, one X chromosome is there, another X chromosome is there. So basically, cyst chooses that which particular X chromosome has to be inactivated. Okay, so in each cell, one of the two X chromosome is randomly chosen to be inactivated, and this inactivation is mediated by the expression of cyst RNA from the selected X chromosome. So basically, the correct answer is exclusively. Okay, now we come to the next question. What is the class of cyst RNA? LNC RNA, shRNA, piRNA, or ncRNA? Think. Right now it is 7:58. I will switch the screen after 2 minutes. Think. We have already studied about it. You have 2 minutes to answer this question. Okay, now I am switching the screen. So the question is, what is the class of cyst RNA? ncRNA, shRNA, piRNA or lncRNA? The correct answer is lncRNA, that is long non-coding RNA. lncRNA. Okay, this is long non-coding RNA, which is approximately, here we have studied somewhere here. Yes, no, here, cyst. Huh, it is 16,000 nucleotides. Okay. Now, can you tell me what defines whether a RNA can be a long non-coding RNA or a normal non-coding RNA? What is the criteria? What is the definition criteria that decides whether it will be a long non-coding RNA or not? Can you tell me? Can you think of a reason? So, long non-coding RNA is an long non-coding RNA which should be greater than 200 nucleotides. If it is greater than 200 nucleotides, then it can be called as a long non-coding RNA. Could you understand this? What I'm trying to say? It should be greater than 200 nucleotides. If it is greater than 200 nucleotides, then that particular long non-coding RNA can be characterized as a long non-coding RNA. And this particular cyst RNA, what is the size? 16,000, which means it is a long non-coding RNA. Okay. Next question is, YY1 influence cyst and X inactivation through A, competition, B, mediation, C, neutralization, and D, suppression. What should be the answer? I'm repeating the question. YY1 influence cyst and X inactivation through A, competition, B, mediation, C, neutralization, or D, suppression. What should be the answer? I'm giving you 2 minutes. After, I'll switch the screen after 2 minutes. Right now it is 8:03. I'll switch at 8:05. Okay. Think. One more minute left. One more minute left. Okay, so now I am switching the screen. Let us see what can be the answer. Hmm. So the question is, YY1 influence ZIST and X inactivation through A. Competition, B. Mediation, C. Neutralization, and D. Suppression. Okay, the correct answer is mediation. So how it can be mediation? First of all, what is the full form of YY1? YY1 is yin yang 1. Okay, YY1 is yin yang 1. It is a transcription factor that mediates the regulation of ZIST expression and plays a role in X chromosome inactivation. So basically, it helps in the expression regulation of ZIST expression. Okay. So what happens? YY1, it basically binds to specific sites which are present at the ZIST promoter, and then it helps to recruit proteins that facilitates the initiation and spreading of ZIST RNA, which is essential for the expression of, or I mean for the inactivation of one of the X chromosome. So the correct answer is mediation. Okay, so what happens is, you should understand that YY1, it binds to specific sites on the ZIST promoter. It binds to specific sites on the ZIST promoter, and it helps to recruit proteins that helps in the initiation and spreading of ZIST RNA. So basically, it is helping in the spreading and initiation of ZIST RNA. And when ZIST is expressed and when ZIST is initiated, your X chromosome inactivation takes place. The correct answer is mediation. Next question is, proposed mechanism of T6 and ZIST interaction is isolation, competition, masking, and euchromatinization. I am giving you 2 minutes to try to answer this question. Think. Right now it is 8, 9. I will switch the screen after 2 minutes. Okay. One minute more. See the question, it is asking what is the proposed mechanism of T6 and cyst interaction? Isolation, competition, masking, and euchromatinization. First of all, you should know that why T6 and cyst interacts with each other. If you know this, then it will be easy for you to answer this question, okay? Okay, now I'm switching the screen. Fine. Okay. So the correct answer is masking. Can you think why masking is the correct answer? Because what is the relation between T6 and cyst? So cyst basically is involved in X chromosome inactivation, that we already know, that cyst is involved in X chromosome inactivation, and T6 is just the opposite of cyst. It has a sequence which is base pairs with cyst, and it inhibits the or regulates the expression of cyst. So basically you should understand that the interaction between T6 and cyst can be described as masking, because T6 binds and prevents the stabilization of cyst RNA. And basically what it is doing is it is actually masking the work of cyst RNA, preventing it from initiating the inactivation process. So if T6 comes and it binds to cyst, the X chromosome inactivation is halted. If it comes and it binds to cyst, then what happens? X chromosome inactivation is halt is halted. So this masking mechanism ensures that the chromosomes expressing T6 remains active while the other undergoes inactivation. Okay, so in this way, this T6 and cyst are kind of antagonist of each other. So I hope this is clear to you. Okay, how this T6 and cyst interacts is T6 is masking the expression of cyst. So basically it cannot undergo the inactivation process, and this masking mechanism, it ensures that the chromosome which express T6 remains active while the other undergoes inactivation. Now the next question is, X inactivation begin from a region which is called as A, T6, B, cyst, C, centromere, and D, XIC. So what is the region from where X chromosome inactivation begins? Right now it is 8:14. I will switch the screen after 2 minutes. Think. One more minute left. Hmm. 30 seconds more. Okay. Now I am switching the screen. So the correct answer is XIC, which is X chromosome inactivation center. So if you see here, let us see. There was a slide which shows about this. Okay. Where was it? Hmm. So the X chromosome inactivation center. So the X chromosome inactivation center basically has all the non-coding RNA such as ZIST, T6, JPX, FTX, and in the X chromosome inactivation center only every process which is related to X inactivation takes place. So the question was, what was the question? No. Asking. Huh. X inactivation begins from a region which is X chromosome inactivation center. In X chromosome inactivation center, only your ZIST is there. In X chromosome inactivation center, only all the non-coding RNAs which are involved in the X chromosome inactivation are present. So, if this is your chromosome, okay, if this is uh, huh, if this is your chromosome, there is a dedicated locus which is meant for X chromosome inactivation, and this locus is known as X chromosome inactivation center, where all the non-coding RNAs, all the long non-coding RNAs which are involved in X chromosome inactivation are present. Okay, so the correct answer is X inactivation begins from a region which is known as XIC, X chromosome inactivation center. Now we'll do some true false. First is histone deacetylase is not required for chromosome compaction. True or false? Think. Right now it is 8:19. I will switch the screen 8:21. Okay. This particular topic also we have dealt in detail in today's class. So you should be able to answer this question. 1 minute more. Hmm. Shall I switch the screen now? So the question is, histone deacetylases is not required for chromosome compaction. The answer is false. Why? We have studied there are two processes. Okay. One is acetylation, the other one methylation. What is the function of acetylation in general and what is the function of methylation in general? Hmm? Acetylation marks the particular chromosome transcriptionally active. It makes transcriptionally active, and methylation marks it transcriptionally inactive. Okay. So histone deacetylases. Deacetylation means acetylation is marking it for activation of the chromosome. So deacetylation is required for the deactivation of chromosome. And if the chromosome is inactive, that means it is compact. Why inactive chromosome is compact? Because if this is a chromosome, it is completely compact, it is not very loosened up, then there is no place for the transcription factors to come and bind and start the transcription process. So you should always remember, in order for the transcription process to start, the chromosome should be loosened up so that there is some place for the transcription factors to come and bind and start the transcription process. So inactivation of chromosome essentially means that the chromosome is compacted now. So deacetylases will remove the acetylation process, which means chromosome will be inactive, which means chromosome will be compact. Here the question says that histone deacetylases is not required for chromosome compaction. So it is false, because histone deacetylases is required for chromosome compaction. Because when histone deacetylase will come, acetyl group is removed. When acetyl group is removed, then the chromosome will become transcriptionally inactive. And when the chromosome will become transcriptionally inactive, but obvious, it will become compact. So the correct answer is false. I hope this is clear to you. Okay, false. Next question is, calico cats are always female. True or false? I'm giving you 2 minutes. Try to answer this question. Right now it is 8:25. I will switch the screen at 8:27. Okay. One more minute left. Okay, now I am switching the screen. So, calico cats are always female. The answer is true. We have studied, we have seen in the example, if you can see here. Where was it? Here. You see, calico cats are almost always female. And what happens here? We have studied what happens in the case of calico cats. It shows, this example describes the random inactivation of X chromosome. So what happens? Coat color. There are two XB and smaller b. This is the dominant one. This is the recessive one. The smaller one codes for black color and this one codes for orange color. Okay. So what will happen if both dominant alleles are present? It is orange in color. If both recessive alleles are present, it will be black in color. But if one is dominant and one is recessive, what should be the color? Ideally, it should be orange color because orange is dominant over black. But it does not happens. What happens is that there are patches if of the coat color. Means there are random patches. Some are orange in color, some are black in color. Why? Because the inactivation of the X chromosome is random. It is not like in all the cells only that particular X which has the dominant allele will be inactivated, or in all the cells all the X which has the recessive allele will be inactivated. No, it is random. In some cells this gets inactivated, so it shows black color. In some cells this gets inactivated, so it shows orange color. So because the inactivation of X chromosome is random, so randomly sometimes the dominant allele is getting inactivated, sometimes the recessive allele is getting inactivated. And because of this, you do not get a whole complete like a orange color, but you get patches. Some are black, some are orange in color. So, and this always mostly 99% it happens in female. So the correct answer is true. So this calico cat is an example that shows random X chromosome inactivation. So I hope this is clear to you. Fine. Next question is, dosage compensation do not cause balance in gene expression. True or false? This is the basic question. I hope that you all know the answer of this question, because this question describes why we are studying dosage compensation at the very first place. Okay. So right now it is 8:30. I will switch the screen after 2 minutes, that is at 8:32. One more minute. Okay, now I'm switching the screen. Okay. Let us see. Not tried. Okay, fine. So the correct answer is false. Why? Because the entire identity or the entire logic of having dosage compensation is to balance gene expression. That is why there is a compensation in the dosage. Why? Because let us take the example of female humans, that females have two X chromosome and males have only one X chromosome. So that means the expression would be twice and compared to males. So in order to balance the gene expression, one of the X chromosome is inactivated, so that there is a compensation that happens. Even though there are two genes of X present, expression is only from one gene. So that is, if the amount of expression here is one, here also it is one, so it is compensated, it is equal. So the entire objective of dosage compensation is to balance gene expression. Okay, so could you understand? So it is all, obviously it is false, because it leads to balance in gene expression. So I hope this is clear. This is the very basics, the very objective of why we are studying about dosage compensation. Okay, now next question, and probably the last question for the day, is Y chromosome can be inactivated by cyst. True or false? I don't know what I haven't opened. It's not the PowerPoint. Anyways, so Y chromosome can be inactivated by cyst. True or false? So right now it is 8:34. I will switch the screen after 2 minutes. That is 8:36. Okay. Okay. Fine. I'm switching the screen now. Hmm. So the question is, Y chromosome can be inactivated by cyst, and the correct answer is false. So why it is false? Because here we are studying about what? Here we are studying about X chromosome inactivation. From since the beginning we are studying about X chromosome inactivation, how X chromosome can be inactivated, what are the different ways of inactivating the X chromosome. We're not talking about the Y chromosome, we're talking about the X chromosome. So Y chromosome cannot be inactivated by cyst. Cyst is specifically designed or expressed from the X chromosome inactivation center, not from the YIC. It is expressed specifically from the XIC, that is X chromosome inactivation center, on the X chromosome, and functions to inactivate one of the two X chromosomes in females to achieve the doses compensation. Okay, the Y chromosome does not have an XIC region, and therefore cyst cannot bind to or silence the Y chromosome. So this is false. Y chromosome, does it have XIC? No. XIC is X chromosome inactivation center. Okay, so therefore the correct answer would be false. So I hope this was clear to you. Now we will quickly, very quickly, within 5 minutes, we'll quickly summarize what we have learned today, and then we'll call it off for the week. So we started with the structure, signal recognition particles, and we studied about the tmRNA. tmRNA is a hybrid molecule which is half tRNA and half mRNA, and how this tmRNA helps in degrading mRNA which is not completely made and detaching the disassociating the ribosome from it, that we have studied. And then we have studied that there are various functions of non-coding RNAs. The non-coding RNAs have regulatory role. For example, they can be used as transcriptional regulators, translational regulators, modulators of protein function, regulators of RNA and protein distribution. And one of the major or most important role of this regulatory non-coding RNA is dosage compensation. So then we studied about what are the different mechanisms of dosage compensation that takes place in mammals, C. elegans and Drosophila. In mammals, there is X chromosome inactivation that takes place. In C. elegans, there is repression that takes place, and in Drosophila, there is hyperactivation that takes place. Okay, this is what we have studied. And then dosage compensation. Then we have studied about the bar bodies. What is the meaning of bar body? Bar body is the inactivated X chromosome. The X chromosome that has been inactivated, that is now stored as bar bodies, that we have studied. And then we have studied how this cyst comes and plays its role. It is a non-coding RNA of about 16,000 nucleotides, and it appears to recruit a specific isoform which is H2A 1.2, and this maintains the chromosome in inactive state. And this cyst also recruit histone deacetylases and methylases, and the activity of cyst is regulated by another long non-coding RNA which is known as T6, and this T6 contains the antisense sequence of cyst, and therefore it base pairs with cyst and it regulates it. Okay, then we come to the calico cat example of how the X chromosome inactivation is random. And then we studied that there can be two types of X chromosome inactivation. First can be imprinted, but this imprinted X chromosome inactivation only takes place at certain stages. And what's that stage? It takes place in trophectoderm and primitive endoderm. And this uh, is basically imprinting of the paternal X chromosome that takes place. And then we have studied about the X chromosome inactivation center and what are the different non-coding RNAs. So basically these non-coding RNAs that like ZIST, T6, JPX, FTX, these are all present in the X chromosome inactivation center and how they help. And then we talked about, then that is what we have majorly talked about today. So basically we have covered the X chromosome inactivation, how the X chromosome inactivation takes place, what is the mechanism, what is the role of long non-coding RNA in the case of X chromosome inactivation, and uh, what are the different types of X chromosome inactivation. It can be your imprinted or random. So all this we have studied. So is it clear to everybody? I hope this is clear. So with this, we'll end today's class. This is what was required. So we have covered week nine's lectures and the questions which are related to week 9. Okay. So now we'll meet again next Tuesday, and in next Tuesday we'll cover the lecture materials and the questions which are related to week 10, whatever topic week 10 has. So I hope this is clear to everybody. Okay, so you can leave now. Okay, so now I will switch off the recording. Just a minute. Hmm.